Hello everyone, welcome. So, we have talked about what are the tools, we have talked about the process of art in a technology. Now we were talking about I need to get the desired product. So, how do I get the desired product? I need bioreactors. So, today we will be talking about bioreactors. So, what is a bioreactor? A bioreactor helps to produce a large volume of culture. The bioreactor is a large vessel where the different cells, such as human or plant or animal cells, can be cultured to obtain new biological products. A bioreactor, it refers to a process, aerobic or anaerobic, aerobic in presence of oxygen, anaerobic in absence of oxygen, where a system supports a biologically active environment. It is chemical process where a system is created for growing organisms such as yeast, bacteria or animal cells in a controlled way. These bioreactors, they are used in industries for creating vaccines and antibodies. I need the product. That is why I am going into the pain of transforming the cells or having a genetic engineering. Coming to the basic structure of a bioreactor, it's a fermenter, if I call it this. It provides a suitable environment to which organisms can efficiently produce a target product that may be cell biomass, a metabolite, or bioconservation products. If I talk about a stirred tank reactor, it will either approximately be a cylindrical or a curved base. So, a curved base will assist it in mixing the reactor contents. The performance of any fermenter, it depends on many factors, but the key is the physical and the chemical parameters that must be controlled after agitation. So, I should have a proper agitation rate, that is the mixing rate. The oxygen transfer has to be optimal. pH, temperature, foam production, all this needs to be maintained. Okay. So if the agitation is more, my cells might get ruptured. Oxygen transfer has to be optimal. More amount of oxygen is difficult. If it gets less amount of oxygen, also the cells would not grow. The pH cannot be, it needs to be optimal. Higher pH or lower pH, acidity of the uh, whole matter needs to be maintained, otherwise the cells would again gets ruptured, or it might shrink off, it might get swell, there might be plasmolysis that might occur. The temperature is also very important, and foam production, too much of foam production would reduce the oxygen which is reaching to the cells. So this is a bioreactor that I would have. So basically there are two kinds of bioreactor, which is simple stirred tank bioreactor, where I have a motor which is going to rotate the blades that I have. There are flat blades. So above everything I have a foam breaker. It will just break the foam up, and the culture growth there, this flat bladed impeller, it will keep on mixing. That is, the motor will rotate, so the blades will rotate. So there's a sterile air which is let inside. So I have an inlet where the acid base for pH control I have one, and for the steam for sterilization. Whereas if I talk about sparged stirred tank bioreactor, here I have a sterile air bubbles which is being sparged inside, which is being let inside. So this increases the surface area of oxygen transfer. So the bubbles, they will increase the oxygen transfer area. So that will be there. So now we have talked about upstreaming, where I have the vector construction, selection of the strain, then I need media optimization is there, then I scale up by using a bioreactor or a fermenter. So there is a fermentation that takes place. After this, whatever product is being produced, I need to purify this and I will have to collect it. This is the downstream processing I will be going ahead in the next video. Thank you.